Let us take some examples that you must have heard. One is Lispro, when lysine is replaced by proline. Lysine by proline. Another one is glue lysine. In this case, glutamic acid is replaced by lysine. And the third one is aspartate. In this case, only the position of aspartic acid changes. Do you know what are the differences between this human insulin and these three new derived insulin by genetic engineering? This human insulin is short acting, while these three insulins are even short acting. That means ultra short acting and even faster. These three new insulins have advantages over regular insulin. Let us understand how. I am writing here three new insulins. We need to take these three insulins just before 20 minutes before taking meal. But regular insulin, in case of regular insulin, we need to take before at least 1 hour. And this is the major advantage of new insulins. Okay. These insulins are more useful in postprandial hypoglycemia. Let us see the beauty of genetic engineering, the synthesis of new drug. One example is there, that is paclitaxel. And the source is yew tree, which is an endangered species. For that, what are you thinking? For the synthesis of paclitaxel, we need to cut the tree? No, because now genetic engineering is there. Synthesis, we need to take a gene from the yew tree and that should be introduced in the cell line, that is bacterial cell line we have taken. In this case, bacterial cell line, example is E. coli, and the product is obtained in this case, that is paclitaxel. Okay, let's revise what we have studied in this class. That what is pharmacology? That pharmacology word that has been derived from two Greek words. One is pharmakon, that means drug. Another one is logia, that means study of. Sorry, I have missed that one. What is the definition of pharmacology? As of we know, it is the study of drugs which interact with the components of a living system that attenuates or decrease. You can also write decrease or inhibit various pathophysiological processes. You need to write this one. Then what is drug? A drug is a chemical with a known structure that alters the pathophysiology for therapeutic system. For example, the chemical we have taken, that is enalapril, that is ACE inhibitor, angiotensin converting enzyme inhibitor, and that affect on the pathophysiology of the body, that is RAS mechanism, renin angiotensin aldosterone system. As a result, the BP decreases, blood pressure decreases. That is the therapeutic effect. After that, we will go to the drug sources, chemical. First one is natural. First one is then classified as the plants. The examples are morphine, digoxin, quinine, reserpine. And the second one is animals, insulin. We have discussed about all these things. And the third one is microorganisms. The popular antibiotics is the example. And in semi-synthetic, we have taken the natural chemical, that is morphine. After acetylation process, diacetyl morphine is formed. After that, synthetic. According to the shape of the receptor or a target, we need to design a drug. That process is known as simply rational drug designing. The drug is never existed before, but we designed it in de novo synthesis. In de novo synthesis, we can just subtly write here de novo synthesis. We design a drug, and just remember, the most common source of drug is the synthetic drug, synthetic chemical. After that, we just go to genetic engineering, where we took a gene and introduced in a cell line and just get the product, which is known as the drug. For an example, we take human insulin. When after introduced in the cell line, we get the desired product, that is in human insulin. We know that is the regular insulin and better use. After extensive use of genetic engineering, we design 3 insulins: Lispro, glulysin, and aspart. How we get this? In Lispro, lysine is just replaced by proline, glutamic acid is replaced by lysine, and the position of aspartic acid we just change. And in shortly you can write these three insulins, these new three insulins are better than a regular insulin, and after that the animal derived insulin, that is from beef and pork. And we need to remember this thing, that the regular insulin, and just remember this thing, change in duration and onset of action is taking place because of which reason. Okay, after that we are going to paclitaxel. The source is U3. How this is done, the gene is introduced into the bacterial cell line, that is E. coli, and the product we get is paclitaxel. So no need to cut the tree to get the anti-cancer drug, that is paclitaxel. We need to remember this thing, that the regular insulin, and just remember this thing, change in duration and onset of action is taking place because of which reason. Okay, after that we are going to paclitaxel. The source is U3. How this is done, the gene is introduced into the bacterial cell line, that is E. coli, and the product we get is paclitaxel. So no need to cut the tree to get the anti-cancer drug, that is paclitaxel. Thank you. How do you like this video? Please comment below. Do like and share, subscribe and click the bell icon to get the notification of the upcoming video. Thank you.